As of 2023, the World Health Organization estimates that 55 million people live with Alzheimer's disease globally, a figure projected to triple by 2050. For decades, pharmaceutical development has been guided by the amyloid cascade hypothesis. The objective has been straightforward: remove the amyloid beta plaques from the brain to halt the disease. But closing the translational gap has proven exceptionally difficult. Clinical trials that successfully clear amyloid beta plaques routinely produce only marginal cognitive benefits for patients. A parallel discrepancy exists in the human population. Neuropathologists have identified a subset of individuals who harbor intermediate to high levels of Alzheimer's pathology, what clinicians classify as Braak stages 3 through 5. Yet these individuals maintain perfectly normal cognition, scoring a 0.5 or lower on the clinical dementia rating scale. This phenomenon is known as cognitive resilience. These two data points force a revision of the standard model. If heavy plaque loads can exist in a fully functioning brain, and removing plaques from a failing brain does not restore function, then amyloid beta cannot be the sole direct executioner of synaptic failure. The model is missing a critical mediating variable. That missing variable is found in the physical architecture of the brain's extracellular space. Surrounding specific inhibitory interneurons throughout the central nervous system are perineuronal nets, or PNNs, specialized condensed extracellular matrix structures that physically ensheath the neuron. This split-screen model illustrates the divergence. Here are two theoretical brains with identical amyloid beta plaque loads. On the left, the brain is actively progressing to dementia. On the right, the individual maintains perfect cognitive resilience. The plaques are equal, but the structural integrity of the tissue is not. The physical integrity of these perineuronal nets acts as the decisive axis of neurodegeneration. The survival or destruction of this matrix determines whether a brain succumbs to dementia or remains cognitively resilient. To understand why the matrix dictates cognitive survival, we must look at these specific cells it protects. PNNs predominantly enclose parvalbumin-positive, or PV+, fast-spiking GABAergic interneurons. These cells operate in a hypermetabolic state. Sustaining high-frequency inhibitory signals puts them under immense continuous operational stress. To survive, the neuron synthesizes physical armor. The PNN's foundation is a hyaluronan backbone. Chondroitin sulfate proteoglycans attach to the scaffold. Finally, tenascin R glycoproteins bind the ends together. This cross-linking pulls the assembly into a dense, rigid lattice covering the cell. This lattice performs specific homeostatic functions. First, the dense mesh physically restricts the lateral mobility of AMPA receptors within the synaptic membrane. By locking these receptors in place, the PNN stabilizes synaptic strength and prevents the uncontrolled rewiring of neural circuits. Second, the protein chains carry a high density of negative charges. This polyanionic field buffers transition metals, specifically chelating iron ions. By sequestering iron, the PNN prevents the generation of reactive hydroxyl radicals, shielding the highly active PV+ neuron from massive oxidative stress. The perineuronal net is an active, necessary biochemical shield. Without this intact extracellular structure, high-frequency inhibitory circuits cannot survive the metabolic demands of normal brain function. When amyloid beta aggregates accumulate in the extracellular space, they trigger pattern recognition receptors on microglia, the brain's resident immune cells. This shifts the microglia from a state of homeostatic surveillance into a highly active, disease-associated phenotype. These activated microglia launch a dual-pronged attack on the perineuronal nets. They secrete matrix metalloproteinases, specifically MMP2 and MMP9, which cleave the core proteins of the lattice. Simultaneously, the microglia initiate direct phagocytic engulfment, physically eating the extracellular mesh. This line graph maps data from the Krapser 2020 study using the 5xAFD Alzheimer's mouse model. As amyloid pathology progresses over 18 months, microglial density rises sharply. In a perfectly mirrored trajectory, the local PNN density plummets. To prove causation, the Krapser team introduced a pharmacological intervention. They administered the CSF1R inhibitor PLX5622, a compound that chronically depletes approximately 90% of the brain's microglia. This bar chart displays the outcome. Removing the microglia, foglia prevents PNN loss. The matrix remains intact and dense, matching the healthy wild-type baseline, despite the fact that the heavy amyloid plaque load is still present in the tissue. The downstream consequences of this localized destruction are severe. Stripped of their ionic buffering shield, the PV plus interneurons succumb to oxidative stress and fail. The loss of these specific inhibitory cells disrupt the excitatory-inhibitory balance of the entire cortical circuit. Microglia are active executioners of the extracellular matrix. By physically stripping the brain of its structural defenses, the immune response forces network collapse long before actual neuronal death occurs. This mechanism aligns precisely with human postmortem data from individuals who exhibited cognitive resilience. A 2024 study by DeVries analyzed frontal cortex tissue across 3 specific cohorts: healthy controls, clinical Alzheimer's patients, and a resilient group that maintained normal cognition despite high Alzheimer's pathology. This plot tracks aggrecan intensity. Compared to controls, both Alzheimer's and resilient groups show a drop in matrix density. Both modify the matrix, but divergence appears in synaptic contacts. Synaptic integrity plummets in Alzheimer's brains. In resilient brains, it remains elevated, matching the healthy baseline. Transcriptomic analysis of the tissue isolates why this happens. The Alzheimer's tissue exhibits a massive upregulation of genes encoding matrix-degrading enzymes, specifically MMP2, MMP9, and several cathepsins. The resilient cohort completely lacks this pathological upregulation. Despite harboring the exact same intermediate to high amyloid pathology, the resilient brains do not mass-produce the enzymes that indiscriminately destroy the matrix. The physical difference between clinical dementia and cognitive resilience comes down to the regulation of the extracellular space. Dementia is characterized by uncontrolled pathological matrix destruction. Resilience is characterized by controlled homeostatic matrix remodeling. Synthesizing these studies yields a unified model. Amyloid beta accumulation triggers microbial activation, the central node where outcomes diverge. Pathologically, microbials secrete uncontrolled MMPs, destroying perineuronal nets and causing dementia. In the resilient route, restricted immune responses allow homeostatic remodeling, preserving PNNs and sustaining cognitive function. Preserving the physical integrity of the extracellular matrix effectively acts as a circuit breaker. It severs the causal link between amyloid pathology and cognitive decline. Engineering future Alzheimer's therapies requires shifting our primary focus towards stabilizing the extracellular matrix to protect existing neural circuits. The first approach utilizes highly selective MMP inhibitors. By blocking specific proteases, we can halt the destructive proteolysis seen in Alzheimer's, while sparing the physiological matrix remodeling required for normal learning. The second approach targets the immune response directly. Rather than the blunt tool of total microglial depletion, the objective is to modulate microglial phenotypes, pushing the cells out of the disease-associated state and back into their homeostatic surveillance roles. The third approach involves manipulating the biochemistry of the matrix itself. Through gene therapy, researchers can alter the sulfation code of chondroitin sulfate, specifically adjusting the ratio of C6s to C4s, locking the matrix into a plasticity-permissive state that defends the neurons it surrounds. The extracellular space functions as a vital active structural defense network. Engineering cognitive resilience means learning to protect this physical matrix.